So let's take a look at Chapter 6 on communication. This is our last topic in Unit 1, and it will still be part of your upcoming unit test. It's a really important chapter because it will form the bridge to our next unit, which is all about the nervous system and communication in general. So let's take a look at what's in this chapter. It's mostly about cell-to-cell communication, signaling pathways, and how the whole body really communicates, not just cell to cell, but from one place of the body to the next. And we're going to take a look at signal transduction pathways as well. So the overview here, cell-to-cell communication. There are two principal ways that physiological signals can be communicated, and that will be electric signals or chemical signals. Electrical signals, fast. That's how your nervous system shoots action potentials. They communicate really fast signals from one place of your body to the next using nerve fibers. Or electric signals can also be going through gap junction channels, where you can then have a direct pathway from cell to cell without using any kind of chemical signals. So electrical signals, fast, nervous system. Chemical signals, also nervous system, but using a chemical. And this chemical is known as a neurotransmitter. In the nervous system, it would be a neurotransmitter. If we talk about the endocrine system, we talk about hormones. There's also something called neuroendocrine pathways. So there's a little bit of overlap between endocrine and nervous system. But nevertheless, chemical signals are those that are using a chemical, whether that be a neurotransmitter or hormone. Both fall in the same category. We also need to take a look at target cells and how they receive a signal and then process that signal. Let's take a look first here at these gap junctions. That's a very fast way for a cell to communicate to the next cell. Basically, it's a pore, it's a channel, and these channels line up from cell to cell. You can see this right here. So this would be a pore that lines up and connects the cytoplasm of one cell to the cytoplasm of the next cell. This happens in the heart, for example, where you have to have very fast signal transduction. Once the SA node fires, you want to get the signal across the entire myocardium really fast, and you're using gap junction channels to allow that very fast flux of the signal. In this case here, these gap junctions, they form what's called intercalated discs in the heart, and you might remember that looking at anatomy slides, looking at intercalated discs. So high concentration of gap junction channels that line up and allow the signal to flow from one cell to the next without hindrance, just making a connection from cytoplasm to cytoplasm, and no receptor necessary, no chemicals necessary, very fast communication. Next here would be a contact-dependent signal. There would be two cells that are communicating to one another. That requires the interaction between membrane molecules on the surface of these two cells. But really what I want to emphasize is this slide right here. So there you would have receptors, and then there's a signaling molecule right here in red. And if the signaling molecule is made by the cell that also has the receptor, then we call that autocrine signaling. So if it's sort of self-stimulation of the cell makes the messenger molecule right here, and then it binds to its own receptor, self-stimulating. Cancer cells unfortunately do that a lot of times. They make their own growth factor and the receptor along with it, and then they self-stimulate their own growth. Another type of signaling that's between neighboring cells, it's called paracrine signaling, and that means that one cell is making the molecule still right here, but the molecule then doesn't bind to the receptor on its own cell, but it binds to the receptor on a neighboring cell, right? Like what you see here, right here. So paracrine signaling is sort of neighborhood signaling, very close proximity of the cells. And now here we have endocrine signaling, and that involves that there's an endocrine cell that is making a messenger. So think of maybe an insulin hormone that is being made by this endocrine cell in the pancreas, and now this endocrine cell puts the hormone that it made into the blood, and now the hormone is traveling in the blood, and it will bind to any cell that has a receptor. If a cell doesn't have a receptor, it's as if the signal doesn't even exist. There's no reception. If the cell has a receptor, then it will pick up that signal, and then there will be a response. So this will lead to a response. It kind of is like your cell phone, and it's kind of funny that the names are so similar, cells and cell phones. So, but think about it. If you are calling somebody, you don't want everybody else's phone to go off. You only want the person's phone that you're calling to go off and receive the signal. So can you imagine what would happen if you dial somebody's number and then 100 people responding, picking up because their phone received the signal too. So it's just like that in human cells, or in general in animal cells. When you're having a hormone, a signal that's being sent out, it needs to bind to a specific receptor that's intended for that particular signaling molecule. And then once that interaction has happened, that the signaling molecule has found its receptor, that's when you get a response in the target cell. So here in this slide, basically the same principle. You have a secretory cell right here that's making some sort of a hormone or chemical messenger in general. And only cell A has the appropriate receptor, that box-shaped receptor, and so only cell A will respond. Cell B and cell C, they just ignore this chemical messenger. They don't have a receptor for it, and so nothing will happen in cell B or C. Only cell A will respond. So continuing here with long-distance communication, i.e. hormones, for example, or here long-distance communication in the nervous system. There you have really fast communication using electrical signals, or you can use chemicals for also long-distance communication, but it's much slower than electrical signals. So neurocrines are chemical signals that are secreted by neurons, and they include neurotransmitter. Most importantly, neurotransmitters are released at axon terminals, and they will do something to the cell that they are synapsed with, so via a synapse. There are also other molecules known as neuromodulators and neurohormones. We'll pick up on those terms later as well, but the most important term here would be neurotransmitters. So here's what that would look like. You have some sort of a neuron, and this electric signal here, that will be communicated via action potentials. I'm going to abbreviate that here with AP, action potentials, very fast communication. Now we're going to reach the end of this axon, and how do we get the signal now across? Here's the target cell. So now we're going to release a neurotransmitter, an NT neurotransmitter. That's the red stuff right here. And it will bind to the receptors that are on the target cell that are very close proximity to the axon terminal. And then this target cell receives this neurotransmitter, and the event of binding to the receptor then triggers a response in the target cell, and there we have the response. Sometimes you have the situation where a neurohormone is released by a neuron into the blood, and then as before, only cells with the receptor, they would respond then to this neurohormone that was released. So neurohormone here from this neuron released into the blood, travels in the blood, only cell with the receptor here will respond. Cytokines, that's another group of signaling molecules. They can act both locally and long distance. They're typically involved a lot with the immune system, so they can communicate between white blood cells. They can mediate stress, inflammation. They have also their role in development and differentiation. Now let's take a look at a generic signaling pathway. So here is the signaling molecule. That's the neurotransmitter or the hormone, and it binds to its receptor. Now, once that interaction has happened, something needs to be communicated to the interior of the cell. And so these intracellular signaling molecules, they form basically a cascade. Think of dominoes that are falling, dominoes that are lined up, and then the first one is falling and it takes down the next one and so forth, until there is a cellular response. And that response can be significant because you can actually trigger various pathways that this could go, and then start different enzyme reactions and make target proteins and whatnot. They can then create a response. So now let's take a look at what type of signaling molecules are there and how do they communicate something to the interior of the cell. I want you to strictly separate water-soluble from lipid-soluble messengers. When you have a lipid-soluble messenger, the receptor that they need to bind to is typically in the nucleus of the cell. So here, let's just take a look at that little circle right there. That's the most important pathway. Here's your little lipid-soluble messenger molecule. That could be a steroid hormone or some other lipid-soluble molecule. And once this lipid-soluble molecule reaches its target cell, there is no need for a cell surface receptor because this thing can just diffuse to the cell membrane. It doesn't need a receptor. Because remember, the cell membrane is phospholipids, and these lipid-soluble messengers, they can freely diffuse through the plasma membrane and even through the nuclear envelope. They don't need any help doing that. And then they bind to receptors in the nucleus. This is really important. So nuclear receptors, and then they bind to the DNA. They become transcription factors. So binding to the DNA, they will turn genes on or off directly in the nucleus. And then the manufacturing of the new proteins, that's what causes the cellular response. So basically, you are changing transcription in the nucleus, and that of course leads to an altered profile of protein products that is being made, and that leads to the cellular response. So one more time, these lipid-soluble messengers, they enter the cell freely, they go into the nucleus even, they bind to nuclear receptors, which are transcription factors, and then they alter gene regulation in the nucleus. Here's another slide that makes it clear. Same thing. So one more time. Hydrophobic signals. These are hydrophobic signals, lipid-soluble stuff. A lot of times these lipid-soluble messengers, they have to be carried in the blood with some sort of a protein. You may have heard of lipoproteins that carry cholesterol. There's these high density, low density, whatnot. Lipoproteins. These are proteins that bind to these lipid-soluble messengers, and for example, a steroid hormone, and then they enable it to be carried in the blood because, well, water-based plasma and the lipid messenger molecules, they wouldn't like to mix, and so you need to somehow transport your lipid messenger in the water-soluble environment of the plasma. Now, that's achieved with these plasma binding proteins. And once that lipid messenger has found its target cell, it will enter the cell freely. It will cross the phospholipid bilayer right here. It will also enter the nuclear envelope freely, no help necessary. And then it will bind to a nuclear receptor. So here, this is the nuclear receptor right here. And in the nucleus, then, the nuclear receptor plus the hormone, they become transcription factors that bind to DNA, and they alter gene expression in the nucleus. Once the mRNAs have been made, then you get new proteins, and that's the response that you see in the cell. So now this cell has responded to this messenger that arrived here. What do we have in this category? All of the steroid hormones plus thyroid hormone work just like this. Okay, so this would be this. In this category here, we have all the steroid hormones, Oops, this does not write very well. But anyway, hormones, I think you get the idea. Plus thyroid